The Professor Jas Shostak at Harvard Medical School developed a similar method. He was awarded the 2009 Nobel Prize for Physiology or Medicine for the discovery of how chromosomes are protected by telomeres. The Professor Shostak has made contribution to the field of genetics. In addition to his contribution to telomere research, he and his co-workers developed a super great idea and published a seminal paper in Nature in 1990. I will show you the Nature paper, very famous in the field. It says, in vitro selection of RNA molecules that bind specific ligands. Today, his method is called in vitro selection method, or SELEX, systematic evolution of ligands by exponential enrichment. In vitro selection, or SELEX, uses successive cycles of RNA selection and amplification. As I mentioned, his technique mainly uses RNA instead of DNA. DNA has double helical structure, which is uniform and stable. RNA is single-stranded, which permits the formation of diverse structures. So let's take a look at a typical protocol of Dr. Shostak's methodology. The firstly, a collection of DNA molecules with different sequences needs to be created. We call this collection a DNA library or DNA pool. The DNA library can be created using a DNA synthesizer. How can you randomize the sequence of synthetic DNAs? You don't want to make these DNAs one by one. You can make them at once. It's really simple. You can simply mix up all the 4 nucleotide components and conduct couplings by DNA synthesizer. But you need to ensure that both ends of these DNAs have defined sequences so that the PCR method can be applied later. In this diagram, red bar shows a randomized DNA sequence, and blue bars indicate defined sequences. This DNA library will then be converted into an RNA library by introducing a special enzyme called transcriptase. Now you have an RNA library. It's ready for selection. Suppose you would like to discover the RNA molecules that binds to protein X. The initial selection is counter selection to remove non-specific RNAs. You use empty beads and mix them with RNA pool. They collect the RNA molecules that do not bind to the empty beads. You can basically collect the flow through. The next, protein X is covalently attached to the beads. They put them on the filter here. Mix them with the RNA pool and wash well. Now, RNA molecules that bind to protein X are separated. They stay on the filter. Only a small portion of RNA library is expected to bind to protein X. So most of them do not bind. So these rare protein X binding RNAs will then be converted into DNA by a special enzyme called reverse transcriptase. You get DNA back. The resulting DNA molecules will be amplified with PCR method. As I mentioned earlier, the original DNA library has defined sequences at both ends. Therefore, the PCR reaction with DNA primers complementary to these sequences would amplify the selected rare DNAs. The amplified DNA can then be deconverted into RNA by transcriptase, this is the end of round 1. The amplified RNA library can be used to start another round of selection and amplification. If you use strict conditions for the selection, the resulting population of protein X binding RNAs will be very, very small. Only a few molecules of RNA may bind to protein X, but do so strongly. After 10 cycles, we will be able to select a certain group of RNAs with extremely strong affinity to protein X. An RNA that binds to a specific target molecule is called an RNA aptamer. RNA aptamers can be a pharmaceutical product. Nextar Pharmaceuticals, a US-based company that has now been acquired by Gilead Sciences, discovered a specific RNA aptamer that binds to vascular endothelial growth factor protein, or VEGF. VEGF stimulates the growth of new blood vessels. The VEGF binding RNA aptamer inhibits the function of VEGF and stops the creation of new blood vessels. Actually, this aptamer can be used to treat patients who have age-related macular degeneration, AMD. AMD is a disease that causes an abnormal proliferation of blood vessels inside the eye, often resulting in loss of vision or blindness. In the United States, the aptamer drug was approved in 2004. Today, this anti-VEGF medicine is being used worldwide to treat patients with AMD. So one of the limitations of RNA-based drugs is that they cannot be taken orally. They need to be injected. The MacGem needs to be injected directly into the eyes. Nevertheless, the RNA aptamer drugs have great potential for the future therapy. Now you have learned how to do in vitro selection of RNA. What kind of RNA aptamers would you like to make? There should be lots of ideas.